Hello, this is Nick Cap, and we are looking at the microbial world. And this is chapters 3 and 4 from your textbook. And I will go over those kind of real quick. Let's see where my pointer is at kind of right now. And with that, we will kind of get going. This is looking at the classification of microorganisms. Okay, the science of classification, especially classification of living organisms, to show the similarities among organisms. And so classification allows us to really kind of look at these organisms and get an idea of kind of how they are related to one another. And when we look at those, we also look at how they are evolved from one another. These different categories we call taxa, and this is the process that we call taxonomy. And we're looking at this, we're studying the evolutionary history of organisms. Number one is to look at kind of what those changes over time. And we have a better idea of when things are related to one another, you have a good idea of what the next thing is and what there is different. We're looking at this thing that we call systemics or phylogeny. And for the classification of organisms, we have the three domains. You were asked to look at the three domains. And we have bacteria and archaea and eukarya. These relate back to the old five kingdom system that we had, which is Monera, Animalia, Plantae, Fungi, and Protista. And microbiologists looking at this in the 70s and 80s kind of really saw that there was quite a lot of difference in there, and that these things that we call Monera really were two vastly different sets of organisms. And again, these are the three domains that we have, and again, the five kingdoms that we have. Our three domains were developed by Carl Woese at the University of Illinois. Yay! Fighting Illini. What we see is he was looking at ribosomes, and he was looking at the 70S and 80S, looking at cell wall, looking at sensitivity, microbiologics. And again, we have this tree of life, and we look at it, you are right here, and so here are the animals that are right there. I'm going to go back here a little bit. And so you're actually more closely related to a plant than actually your gram-positives are to Thermococcus. So there's big differences that we're looking at here, and the tree of life is very important. Again, there are two major types of cells, one we call prokaryotic cells and eukaryotic cells. In the microbiology class, we will be actually looking at kind of both of these. The prokaryotic cell have one circular chromosomes that's not enclosed by a membrane, no membrane enclosed organelles, 70S ribosomes, and they reproduce by what we call binary fission, is that basically they kind of break in half. Multiple eukaryotes have multiple chromosomes. These are encased by a nuclear membrane, separated. These are membrane-bound organelles, 80S ribosomes, and they divide by a process that we call mitosis. Let's get going on here. So there are a number of things in common, and you will be responsible for kind of looking at these, and these are the two types of cells that we look at. And I will leave you, I won't necessarily go over those, I'll leave you to kind of look at those. So we have some similarities and we have some differences. Lynn Margolis in 1967 provided what we call the endosymbiotic theory, where eukaryotic cells evolve from prokaryotic cells. And so what we see here is we have this prokaryotic cell, and this prokaryotic cell engulfs some other prokaryotic cells, and rather than killing those and eating those, if it was able to enslave those cells or symbiose those cells, then it would be able to do kind of more things. And we kind of see this. And so this modern heterotrophic eukaryote came from those organisms. Again, this is based on the endosymbiotic theory is based on some materials, and we'll have you in class kind of fill this out. We're looking at the prokaryotic cell, eukaryotic cell, and the eukaryotic organelles, mitochondria. What's their DNA look like? What do the ribosomes look like? What does the growth look like? Phylogenetics is basically looking at that each species retains some characteristics of its ancestors. And so again, when you have a species originate another microorganism, that microorganism is going to be like, it can't be totally brand new. And so these grouping of organisms occurred by looking at the anatomy. And again, when we thought anatomy was the most important thing, we separate things according to that. And so when we look at the fossil record, we see how different these things are, but they're really not all that different. Ribosomal RNA was also something that we look at. Our taxonomic hierarchy is that a species, every member of the species is related to one another. In a genus, we have several species in a genus, several genus in a family, several orders in a family, and these are kind of all related. So you can see the relationship of how things are related to one another as we kind of go through this. Classic example that we get is the species Homo sapiens. Our genus is Homo, hominids with upright posture and large brains. Hominids are primates with relatively flat faces and three-dimensional vision. Order primates, mammals with collarbones and grasping fingers. Mammals, chordates with fur and hair on milk. Chordates. So basically, these all kind of relate to one another. And again, we are animals, but we are a specific type of animals, and we are related to these things that are close to us. And so we have the domain Archaea. Now, classification of prokaryotes is a little bit different. Prokaryotic species is a population of cells with similar characteristics. The culture of bacteria are given time in a media and able to grow. When we clone is a population of cells derived from a single cell. A strain is genetically different cells within a clone. And again, here we'll be using this as a classic example of streaking for a single plate. That's where we take some bacteria on a loop, we put it on a loop, and then we see this here. We could grow this in a liquid culture, which we will be doing in a laboratory. Classification of eukaryotes. Again, Animalia are multiple no-cell walls. They're heterotrophic. Plantae are multicellulars that have cell walls, and fungi are unicellular to multicellular. They develop from spores and hyphae filaments. Protista, this is basically a catch-all kingdom of eukaryotic organisms that fit in other kingdoms. Basically, they're single-celled, and they may move and stuff like that. Groups are based in the clades based on their ribosomal RNA. And so, we look at these things called classification for a second. Classification is placing an organism as a group-related species. Identification is matching an organism of unknown to known organisms. And again, we're looking at clinical lab identification. And then classification, we're looking at characteristics of known organisms. We use a number of different ways to determine this. We have Berge's Manual Determinative Bacteriology, which provides identification schemes for identifying bacteria. Berge's Manual of Systemic Bacteriology provides a phylogenetic information based on bacteria and archaea. And this is based on RNA sequencing. And again, these are starting to kind of meld into one another now. And in past, and when we've done things in the lab, when we're in the lab, we determine this a little bit more. But in the future, what we're going to be looking at is this kind of systemic RNA-based news. We have a number of identification methods: morphological, differential staining, our gram stain, acid fast, capsule stain, endospore stain, and biochemical test. And we're kind of looking at genetics. And so right now what I'm going to go over is kind of what microorganisms should look like. We have the cocci, which are the circles, bacilli, rods, vibrio, coccobacillus, which are small rods, and spirillum and spirochetes. And again, you're going to need to know these. Also, their arrangement, how are they clustered together? Single cocci, diplococci, staphylococci, streptococci. Again, we've seen those before. We gotta share present. Sorry about that. I didn't want this bar to go in here. Let's get rid of that bar. But we see that's there. So differential staining is looking at the gram stain. Main steps of the gram stain: the heat smear, decolorizing, and these are all the steps that we kind of go through here, and we will talk about those steps, and you will list those steps. And so class activity, we're going to do a little bit of this. What is the purpose of making a heat fix smear? And we're going to go over this in class. The mechanism behind the gram stain. And so what we tend to see is our gram positives and our gram negatives. Our gram positive tend to have a very large unit structure of peptidoglycan, and our gram negatives have a relatively small unit of that peptidoglycan. And these different structures are in their cell wall, and this is what we call a peptidoglycan. And so again, let's think about it. Bacteria number one is gram negative. However, many gram stains showing purple. I know I used a bacteria culture. What can be wrong in this procedure? And so here we're looking at some critical thinking. What can be going wrong with this material in our critical thinking? Here's identification methods: our differential staining, acid and fast staining. This here is kind of looking at bacteria that cause tuberculosis, Mycobacterium and Nocardia. Differential staining, what we're doing is we're looking for structures, and so this is looking at your capsule stain. Differential staining is also looking for your spores and bacillus. Biochemical test can determine the presence or absence of different bacterial enzymes. And so basically, in reality, we're all kind of looking at genes inside of there. But again, before it was very fashionable or very possible to do genetic determination of bacteria, we had to look at these biochemical tests. And again, we can look at which enzymes of the bacteria will be determined by basically catalyzed biological reactions, metabolism, the endoenzyme, function of the cell wall, and exoenzyme is outside of the cell wall. And so lastly, the one that we're looking at very big now is looking at genetics. And in genetics, we're looking at DNA fingerprinting. We use DNA restriction enzyme analysis, polymerase chain reaction, nucleic acid hybridization, fluorescent in situ hybridization. And so DNA fingerprinting is looking at something like this here. We're looking at kind of what that DNA looks like and what these are. And you can tell what those are. So number 3 is this unknown. What is this number 3? And it looks similar to that. Number 5 is unknown. Number 2, Citrobella salmonella, and so we can identify kind of what these are with some pretty specific extracts. And so that's it for chapters 3 and 4 in your lecture. This has been general microbiology, and I hope you had a good time with it.